So for decades, there was this really fascinating medical paradox that just completely baffled scientists. And I mean, it basically goes like this. Let's say a surgeon transplanted a piece of foreign tissue into your kidney or, grafted it onto your skin.
Your body would just freak out.
Right. The immune system sounds the alarms almost instantly. It recognizes the foreign invader, attacks it, and you get immediate rejection. But if that exact same surgeon took that exact same foreign tissue and transplanted it into your brain...
Nothing.
Exactly. Nothing happens. It just sits there. The immune system, it doesn't swarm it. It suffers what researchers called a delayed rejection, like sometimes taking weeks or even months to finally break down.
Yeah, and because of that single glaring observation, this massive dogmatic belief was born. I mean, it dictated medicine for the better part of a century. We thought the central nervous system, so your brain and your spinal cord, was this immune-privileged fortress.
The completely sealed vault.
Right, locked away behind this impenetrable wall, totally isolated from the messy chaos of the body's normal immune system.
And, I mean, it's pretty easy to see why that theory held on for so long. We love clean categories,
Oh, for sure. We want to believe the immune system handles all the dirty work of the body, and the brain just sits high up in its ivory tower handling the mind. The idea of the brain as this isolated, sterile sanctuary is just, well, it's incredibly comforting.
Comforting, yes, but also completely wrong.
So wrong.
Which is exactly why we are jumping into our deep dive today. We're looking at a massive new review article from the journal Cell, Death, and Disease. And our mission today is to completely tear down that fortress myth.
Yeah, because the brain is not a sealed vault at all.
Not even close. It's this highly dynamic, beautifully orchestrated immunological interface. I mean, it's constantly talking to your gut, piping immune cells through solid bone and flushing out trash while you sleep. But to understand how all of this works, we really need to start at the boundaries. Right. If the brain isn't a locked fortress, how does it actually keep the general riffraff out? How does a fortress actually operate more like a bustling, highly exclusive nightclub?
The nightclub analogy actually holds up incredibly well, especially when we look at our first major checkpoint, which is the blood-brain barrier.
The famous BBB.
Exactly.
Yeah.
For the longest time, the blood-brain barrier was taught as a literal static brick wall. But when you look at it under modern electron microscopy, you see this living, breathing filter.
Okay, so let's unpack that. What makes up the filter?
Well, the foundation is made of endothelial cells. These are the cells lining the inside of your blood vessels. And in the brain, they are stitched together by these incredibly dense proteins called tight junctions.
So if we're using the nightclub analogy, those tight junctions are like the heavy deadbolts on the front doors?
Yes, exactly. But those endothelial cells aren't just working alone. They have a ton of backup.
The bouncers.
The bouncers, right. Wrapping around the outside of those blood vessels are the end feet of these star-shaped cells called astrocytes, along with these structural support cells known as parasites. Together, they form this cooperative security team. They don't just blindly block everything. They are highly selective about who gets a VIP pass.
So let's look at how that security team actually operates during a crisis. Let's say there's a localized infection in the brain tissue.
A bacterial breach or something.
Right. You need white blood cells to get in there and fight it, but you can't just open the floodgates. So the endothelial cells actually pull out their molecular ID scanners.
Yes, the adhesion molecules.
Right, the adhesion molecules. And instead of giving everyone listening a massive alphabet soup of acronyms, let's just focus on one really important one, which is ICAM1.
That's a huge one.
When there's trouble, the cells lining the blood vessel express this protein, ICAM1, on their surface, and it basically acts like microscopic Velcro.
Yeah, that's a great way to visualize it. When a specialized immune cell floats by in the bloodstream, it catches on this Velcro. It physically rolls to a stop, sticks to the wall, and then, and this is an amazing mechanical feat, it literally squeezes itself between the tight junctions and slips into the brain tissue.
But wait, how does it know to squeeze in right there?
That squeezing process is heavily regulated by these chemical breadcrumbs called chemokines.
Ah, okay.
So if the adhesion molecules are the ID scanners, the chemokines are the VIP invites. The brain cells secrete these chemicals to create a literal scent trail. It ensures that only the specific, tailor-made immune cells needed for that exact crisis are guided in.
But I guess my question is, why be so restrictive? If there's an infection, wouldn't you want a massive immune response? Bring it, everybody.
Absolutely not. And that is the core of why the brain's immune landscape is so unique. You have to remember, the brain is housed in a rigid, bony box. your skull.
Right, there's no room to expand.
Exactly. If you let every immune cell rush in at once, you trigger a massive inflammatory cascade. Inflammation brings fluid. Fluid brings swelling. Like in your arm, a swollen cut is annoying. But inside the skull, swelling has nowhere to go.
It just crushes everything.
It crushes delicate neurons, it cuts off the blood supply, and it causes permanent catastrophic tissue damage. The bouncers have to maintain strict order to prevent a complete riot.
Wow, okay, that makes total sense. So let's do a really quick recap of this first part for everyone listening. Bullet 1. The blood-brain barrier isn't a wall, it's a dynamic checkpoint with strict bouncers.
Using ID scanners and VIP passes.
Exactly. But here's the thing. Any busy nightclub generates a ton of trash, right? Empty glasses, sticky floors. For the longest time, I remember reading that the brain had no lymphatic system to drain waste. So how is it taking out the trash?
Well, it actually has its own specialized plumbing, which we now call the glymphatic system.
Glymphatic with a G.
Right. Remember those astrocytes we just mentioned, the bouncers with the end feet supporting the barrier?
Yeah.
Those exact same end feet are packed with specialized water channels called aquaporin 4.
Okay, let's break down how that mechanism actually works, because it's brilliant. These aquaporin 4 channels act like tiny pumps, right?
Exactly. They take cerebrospinal fluid, which is that clear liquid bathing the brain, and they physically pump it deep through the brain tissue.
It's literally like turning on a pressure washer. This flow just washes away the metabolic waste, the dead cell debris, and specifically the misfolded proteins that build up during the day.
Including beta amyloid, which is the primary protein implicated in Alzheimer's disease. The glymphatic flow pushes all of this debris into the spaces around your veins so it can be cleared out.
And here is the detail that completely blew my mind in the source material. This internal washing machine doesn't just run at a constant speed all day. It is up to 60% more active while you are in deep sleep.
The slow wave sleep phase specifically.
Yes, because during deep sleep, your actual brain cells mechanically shrink.
It's incredible.
They physically contract slightly, which widens the spaces between the cells, allowing this fluid to rush through like a flash flood. I love the mnemonic for this. Think of it as the AAA night shift. Astrocytes use aquaporins to clear amyloid.
The AAA night shift, that's perfect. And it's vital to understand that this system isn't just taking out the garbage, it's also clearing out antigens.
Which are what exactly?
Antigens are the molecular fragments of viruses, bacteria, or damaged cells that the immune system needs to essentially see in order to know if there's a threat. If that fluid flow is weak, those antigens get trapped, and you fundamentally alter the immune environment of the brain.
Which really brings me to a question I want to throw to you, the listener. Think about your own sleep habits. Based on this AAA night shift, how might a bad night's sleep be physically trapping toxic proteins and altering the immune environment in your brain right now? It's pretty terrifying.
It is a sobering realization, for sure.
Yeah.
But it also presents this massive anatomical mystery.
Oh, right, because where does the trash go?
Exactly. If the glymphatic system is pressure washing all this waste and these antigens away from the deep brain tissue, where does it go? You just pointed out that the skull is a sealed box. And up until about 2015, medical textbooks explicitly stated there were no lymphatic vessels in the central nervous system to carry fluid away.
Which brings us to one of the biggest paradigm shifts in modern neuroscience.
It really was. In 2015, researchers finally found functional lymphatic vessels hidden right in the dura mater, which is that tough, leathery outer membrane wrapping around the brain.
So the brain actually has a drain.
It does. They proved these vessels existed by looking for very specific protein markers, like LIVE1 and PROX1, that only lymphatic cells carry. This was an absolute rewrite of human anatomy.
That's wild. So these meningeal lymphatics catch that pressure-washed fluid, the waste, the antigens, and they pipe it all straight down into the deep cervical lymph nodes in your neck.
Exactly. So the brain is actively exporting its intelligence, the antigens, down to the body's main immune headquarters in the neck.
But the review goes even further than that, which is crazy. It highlights that the dural sinuses, these large venous channels running through the brain's lining, aren't just passive drain pipes, they are highly organized security checkpoints.
Oh yeah, they are packed with antigen-presenting cells, or APCs.
Let's unpack what an APC actually does for a second. Their entire job is to capture foreign invaders, chop them up into little recognizable pieces, and hold those pieces up on their surface. It's the biological equivalent of holding up a wanted poster.
That's exactly what it is.
And roaming T cells, which are basically the immune system's assassins, are constantly patrolling these dural sinuses, just looking at the posters. So the brain isn't isolated at all. It has a dedicated built-in border patrol right in its lining.
It does, but the most surprising part is actually where those Border Patrol guards are coming from.
Right.
For a century, we assumed any immune cells operating near the brain had to travel there through the general bloodstream, from the body's main bone marrow in the pelvis or the long bones. But recent research uncovered something almost unbelievable.
The secret tunnels.
The secret tunnels.
Okay, I have to push back here because I have literally held a human skull model. It is solid, dense bone. What do you mean there are secret tunnels? How are cells tunneling through bone without us noticing until just now?
I know, it sounds like complete science fiction. But using really advanced tissue clearing and high-resolution imaging, researchers discovered microscopic ossified channels. They physically connect the bone marrow inside your skull directly to the duramater underneath.
So the skull is not just a protective helmet, it's an active military barracks.
Precisely. It is deploying troops, specifically neutrophils, B cells, monocytes, directly into the brain's borders through these microscopic bone tunnels, completely bypassing the general bloodstream.
Bypassing the blood entirely, that is wild.
And what's fascinating here is that we know this drives actual disease pathology. The researchers proved this using the standard animal model for multiple sclerosis, which is known as the EAE mouse model.
Okay, how did they prove it?
They fluorescently tag the immune cells in the skull's bone marrow with one color, and tag the cells in the rest of the body's blood supply with a different color.
Oh, that's smart.
Right. So when they induced the MS-like brain injury, they just watched to see where the responding immune cells came from. And the vast majority of the cells invading the central nervous system marched right down those secret tunnels from the local skull bone marrow.
Not from the blood.
Not from the blood. The skull bone marrow acts as a highly specific, localized immunological reservoir just for the brain. The immune cells born in your skull are actually genetically distinct from the ones in your leg. They are tailor-made for the brain's unique, delicate environment.
Okay, time for our second quick recap. Bullet 1. The lymphatics in the meninges drain waste directly to the neck. Bullet 2. The skull has secret tunnels piping immune cells straight into the brain's lining.
Nailed it.
Which completely changes how we should think about head trauma. I want to pose this to the listeners. If your skull is physically piping immune cells directly into your brain's lining, how does that change the way you view a concussion or even a brain tumor? When you bump your head hard, you aren't just impacting inert bone. You are physically shaking up the brain's private immune factory.
You're altering the barracks.
Yeah.
And that localized view is essential because once we move past the borders, past the meninges and the dural sinuses and go deep into the actual brain tissue, the parenchyma, we meet the local guardians. The resident immune cells.
The famous microglia.
Chief among them, yes, the microglia.
And the microglia are fascinating because of their origin story. Like, they don't come from your bone marrow as an adult.
No, they don't.
They actually migrate into the brain tissue from the embryonic yolk sac when you are just a tiny developing fetus, and then they stay locked in that brain compartment for your entire life.
And they are incredibly busy. In a healthy brain, we used to call them resting microglia, but honestly, that's a terrible term. They are highly modal.
They're constantly moving.
Yeah, they have these long, spindly tendrils that are constantly extending and retracting, physically touching the synapses and probing the brain tissue for any sign of damage or viral invasion.
And when they find damage, they don't just act like garbage disposals eating the debris, right? They can actually transform into those antigen-presenting cells we talked about earlier. They upregulate molecules on their surface, specifically MHC class II, which allows them to hold up those wanted posters locally, directly activating any T cells that manage to cross the blood-brain barrier.
But the microglia don't act alone. They work in tandem with our old friends, the astrocytes.
So bouncers.
Exactly. But inside the tissue, astrocytes are essentially the bipolar regulators of the brain's immune tone. Depending on what the microglia tell them, astrocytes can flip a switch and release pro-inflammatory alarms.
Like IL-1 beta, TNF-alpha.
Yes, these cytokines chemical messengers that basically scream, we have a breach, bring in the troops and start the inflammation.
But they can also do the exact opposite, right? Astrocytes can release anti-inflammatory healing signals like IL-10 or TGF-beta.
And the mechanism there is so crucial. What TGF-beta does is induce the creation of regulatory T cells, or TREGs.
Okay, TREGs, let's define them.
If normal T cells are the assassins, TREGs are the peacekeepers. Their entire biological purpose is to suppress runaway inflammation so the immune system doesn't accidentally destroy healthy brain tissue while fighting off an infection. Your cognitive health absolutely relies on this delicate seesaw between the pro-inflammatory alarms and the anti-inflammatory peacekeepers.
And here's the truly incredible part. The entity holding the remote control to that delicate seesaw, the thing deciding if you get alarms or peacekeepers, is your gut.
Yes, the gut-brain axis.
I know gut-brain axis is a massive buzzword right now, but the mechanics laid out in this review are just stunning. Like, how does bacteria in my large intestine physically control the microglia inside my skull?
Well, we can clearly see the connection when we study germ-free mice. These are mice raised in sterile bubbles with absolutely no microbiome.
No bacteria at all.
Right. And these mice have severely defective microglia. The tendrils are stunted, they don't mature properly, and they wildly overreact to minor stress. And the reason is they are missing vital metabolites produced by normal, healthy gut bacteria.
Specifically, molecules like short-chain fatty acids or SCFAs.
Exactly.
When you eat dietary fiber, you can't actually digest it yourself, but the bacteria in your gut ferment that fiber and produce these SCFAs. These tiny molecules are absorbed into your bloodstream, they travel all the way up to your head, cross the blood-brain barrier, and physically bind to receptors on the microglia.
They literally alter the gene expression of the brain's immune cells.
It's an invisible chemical signal. Those short-chain fatty acids act like an infrared beam from a remote control, continuously programming the microglia to stay calm and mature, while also promoting the creation of those peacekeeping Treg cells.
It is the ultimate example of systemic integration. The brain is locally protected, but globally influenced.
Okay, third recap time. Bullet 1. Microglia and astrocytes balance inflammation and repair locally. Bullet 2. Their maturity is heavily programmed by gut bacteria metabolites like SCFAs.
Spot on.
So, question for you listening. What did you feed your gut microbiome today? And how might that exact meal be dictating how your microglia respond to stress right this second? It completely reframes how we think about eating.
It really does. And, we really need to look at what happens when this beautifully integrated system breaks down.
Yeah, that's where the rubber meets the road. All this regulated surveillance is great until the seesaw tips too far. The review highlights 3 specific diseases that show us exactly how the interface fails. Let's look at multiple sclerosis first.
Okay, so multiple sclerosis is the classic example of the border failing to keep the peace. In MS, autoreactive T cells, which are immune cells that have mistakenly learned to view myelin, the protective coating on your neurons, as an enemy, are primed somewhere out in the peripheral body. They travel up to the brain, and they hijack those adhesion molecules on the blood-brain barrier we talked about earlier.
The VIP ID scanners.
Right. They squeeze through the tight junctions and launch a devastating inflammatory attack on the healthy brain tissue.
But the silver lining here is that understanding this mechanism actually gave us a way to fight back. Modern MS therapies, like the drug natalizumab or ocrelizumab, work precisely by targeting the blood-brain barrier or depleting certain cells. Natalizumab physically binds to and blocks those adhesion molecules.
It literally locks the bouncer's door.
Exactly. It locks the door so the rogue T cells can't stick to the wall and get in. The very fact that we can treat a brain disease by altering the immune system in the systemic bloodstream proves that the brain is not an isolated fortress. It has doors, and we can lock them.
Yeah, let's contrast that with Alzheimer's disease. If MS is an invasion from the outside, Alzheimer's is a tragedy from the inside. It's a failure of the local guardians.
The micro.
Yes. We talked about how the glymphatic system clears beta amyloid proteins. Well, when that pressure washer system fails, maybe due to chronic poor sleep or aging amyloid plaques begin to build up, early on, the microglia actually try to help.
They try to clean it up.
Right. Using a specific genetic receptor called TREM2, they recognize the plaques, engulf them, and try to physically clear them away. In this early phase, the microglia are highly protective and anti-inflammatory.
But they just can't keep up. Over years and years of chronic exposure to these toxic plaques, the microglia get completely overwhelmed, and mechanically, they undergo this phenotypic flip.
They change their entire behavior.
Right. They abandon their anti-inflammatory cleanup duties and mutate into a toxic hyperinflammatory state. They start pumping out reactive oxygen species, which is essentially cellular bleach, and sounding those cytokine alarms. Instead of clearing the disease, the overwhelmed microglia end up driving the neurodegeneration. They end up burning down the house just to kill the spider.
Which brings us to the third and honestly perhaps most insidious kind of failure, gliomas, or brain tumors. Because if MS is the immune system attacking too much, And Alzheimer's is the immune system getting exhausted. Gliomas represent the dark side of the brain's desperate need for peacekeepers.
Tumors are essentially biological hackers, right? They hijack the brain's innate desire to suppress inflammation.
Precisely. A glioma doesn't just grow. It actively manipulates its environment. The tumor secretes chemical factors that heavily recruit those regulatory T cells, the TREGs we talked about, along with other suppressive cells called MDSCs. It pulls the peacekeepers in tight, creating a massive, highly effective suppressive shield around the cancer.
But it doesn't stop at building a shield. The tumor actively disarms any T cells that do manage to break through to attack it.
Yeah, this is the really devious part.
It does this by forcing the attacking T cells to trigger inhibitory receptors on their own surface, specifically receptors called PD-1 and CTLA-4.
The brake pedals.
Right. These are essentially the T cell's natural brake pedals. The tumor steps on the brakes and the T cell becomes completely dysfunctional and exhausted. It just shuts down, leaving the tumor free to grow.
And this is exactly why the absolute frontier for treating brain cancer right now is immunotherapy. We aren't just relying on radiation or chemo anymore. Drugs called checkpoint inhibitors are designed to specifically block PD-1 and CTLA-4.
They physically cut the brake lines on the T cells.
Exactly, waking them back up from their exhausted state.
And therapies like CAR T cells take it a step further. We can actually extract a patient's T cells, engineer them in a lab to completely ignore the tumor's suppressive shield, and infuse them back in so they can assassinate the cancer. We are fighting brain cancer not by suppressing the immune system, but by turning it back on.
It is a complete inversion of the old dogma.
It really is. Okay, final recap before we wrap up. Bullet 1. Neurological disease occurs when this delicate balance tips. Either failing to stop an attack, like in MS, failing to clear a waste, like in Alzheimer's, or being suppressed by invaders, like in gliomas.
And if we synthesize everything we've explored today from this review, the overarching takeaway is undeniable. The concept of absolute immune privilege is dead. The central nervous system is not a walled garden. It is a site of finely calibrated, dynamic immunological engagement.
Beautifully said.
The lymphatic drainage into the neck, the secret tunnels in the skull bone marrow, the remote control of the gut microbiome, they are all inextricably linked parts of a single continuous system.
And that leads me to a final mind-expanding concept pulled from the source material that I really want you to take away today. In this continuous system, there's a phrase: compartment rules matter.
Compartment rules.
We've learned today that the tough outer meningeas, the fluid-producing spaces, and the deep brain tissue all obey totally different immune logics. They deploy different cells, they have different chemical rules, and different tolerances for inflammation.
Which raises a massive question for the future of medicine. I mean, right now, if you have dangerous brain inflammation, a doctor gives you a systemic immunosuppressant, a blunt instrument that affects your entire body, leaving you vulnerable to infections everywhere.
But if we truly understand these different compartments, the future of neurology is precision targeting. Imagine a future where we don't have to use that blunt instrument. What if we can tune the specific immune response of just your cerebrospinal fluid, or just your skull's bone marrow barracks, leaving the rest of the brain's delicate networks and the rest of your body completely untouched?
That is the promise of this new paradigm. We are learning how to harness the immune system's power to heal the brain, on the brain's own highly specific terms.
So what does this all mean for you in your daily life? The next time you wake up with a bit of brain fog or feel overwhelmed by stress, I want you to remember that your brain isn't sitting passively in a static vault. Ask yourself, is it my lack of deep, slow-wave sleep backing up the pressure washer? Am I starving my gut microbiome of the fiber it needs to send calming signals to my microglia? Or are my brain's border guards just working overtime? Your brain is a bustling, highly regulated nightclub, and everything you do determines who gets past the velvet rope. Keep diving deep, and we'll catch you next time.